Hello everyone. Thank you for staying so late for this last session. So let's get started. My name is Sean. I am co-chair of the Unique Student Affairs Committee, and I am joined by...
I'm Charlotte, the other co-chair of the committee.
Yeah, and so, again, thank you very much for being here for the lightning presentations, where we are trying to showcase student work from the Unique community. And so, without further ado, let's get to our first 8 talks today. There'll be 8 talks tomorrow as well. And so, if you'll join me, what's the word I'm looking for? Catherine Bouchard is the name of the person who shall be presenting, irrespective of the word. Welcoming, join me in welcoming Catherine Bouchard.
Okay. Hi, so I'm Catherine, breaking the ice. I'm from a PhD student from Université Laval. I'm presenting my poster number 6, so you might recognize the black background. We're kind of standing out with the black background poster, so make sure that if you have any question, you can come and see me during the numerous poster sessions, poster number 6. So during my PhD, most of the projects I've been working on, the objective was to increase what we can do with the images we acquire, because we're always stuck in between different compromises, between the number of channels we can acquire, the temporal resolution, the spatial resolution. So what I aim at doing is to try to use deep learning to kind of bend this space of compromise and get as much information as possible from the acquisitions we do. So for this specific project, I was trying to multiplex images, so that means try to image as many different channels as possible. So in fluorescence microscopy, a channel is a protein, so we're not just making beautiful images with many colors, we're actually looking at more proteins the more channels we have. So what we've been using is fluorescence lifetime microscopy, or FLIM. So what this does, it's a microscopy technique that doesn't just count the number of photons we acquire at each pixel position, we also count the time it takes for the pixel to reach the detector. And this is a photophysical property of each emitter, so if we use different dyes, we are going to get either longer lifetime values or shorter values. So this can help us identify what emitter emitted the photon. The problem with these techniques is that it requires a lot of photons at every single pixel. But when we're working with neurons, you might know that they're very fragile to fluorescence. So we cannot get as many photons as we need to be able to multiplex the images. Also, we need to use a super high spatial resolution, so super resolution. So for example, with STED, we limit the effective emission. So that's another aspect that makes it so that we have less photons that we acquire. So the method we've developed is called latent unmixing. So what it does is it takes as input a volume of shape x, y, and t, where t is the lifetime of the photons. And it processes all these dimensions at once to try to project from the input space, where all the exponential distributions are overlapping, to a different space where they are forced to be separated, so that we can use bandpass filters in this space and retrieve the abundance of every single emitter in every single photon. So the intuition here is that by using a 3D unit, we can process all the dimensions at once, so we can combine the information from adjacent pixels to kind of simulate a context where we would have more photons than what we actually have. So I have a lot of results to show, so if you want to come see them at my poster, but these are the key results. The first one being that we can unmix up to 4 channels, so we can image up to 4 proteins at once, which was not achievable with the state-of-the-art, which is the phasor method that we show here at the bottom right. The second key result is that we can train on simulated data and still apply it to real FLIM measurements. So this really lowers the hurdle of having to acquire super big data sets, super complex data, to be able to train deep models. And finally, we show that we are more robust than the state-of-the-art for both the distance between the lifetime of 2 emitters, so we can use dyes that have closer lifetime values, which opens up new possibilities for combinations of dyes. And we're also much more robust to low photon counts, so even if we're in difficult imaging conditions with neurons, we can still unmix the images using our method. So this is a preprint for now, but you can scan the QR code or come see me at my poster. Thank you.
Excellent. And if you'll join me in welcoming our next presenter, Maxime Daigle.
All right. So the hippocampus is known for its critical role in long-term memory, but it's also known for its spatially tuned cells. And the discovery of those neurons created the hypothesis that the hippocampus could function as a spatial map, where the different spatially tuned neurons would encode spatial information about different spatial location. But then what was learned is that the hippocampal neurons frequently encode mixture of features that can be both spatial and non-spatial, and that the non-spatially tuned neurons, the non-spatially tuned neurons still contribute to spatial encoding, and that the single cells tuning only weakly predicts the importance of those neurons in spatial encoding. So that raised the question about what is the function of those spatial tuning if it's not about spatial encoding, why some neurons are spatially tuned and others are not. And so to investigate that, we have neural recording from macaque monkeys' hippocampus while they use a joystick to navigate a virtual maze and perform an associative memory task. And then we built a linear model that contained all the features that are relevant in this task and predicted the neural responses from them. And what we see is that this ideal observer is able to significantly better predict the neural responses than a model that only contains spatial information. But this model is limited by a linear combination of features. So next we train deep reinforcement learning models to perform the same task as the monkey, except in the simplified 2D environment. And so both the model and the animals both receive egocentric visual input, aim to navigate to the goal that gives the best reward, and then to be able to do that, need to learn the association between goal and cue objects. And what we found is that the prediction from the activation of those layers are even significantly better at predicting the neural responses of hippocampal neurons. But what is really interesting about this is that this improvement is not uniform across all neurons. What we see is that the simple linear combination of features is actually pretty good at predicting the responses of spatially tuned neurons, and that the added complexity of one is provides limited improvement. But for non-spatially tuned neurons, this added complexity is really required to predict the responses of that subset of neurons. And so there's a lot of difference between those two models. So the next question is, can we pinpoint the exact difference that enable the prediction of that subset of neurons? And what we see is that we can change a single layer, the recurrent layer, from a nonlinear recurrence to a linear recurrence. And the model is still able to perform the behavioral task exactly the same, except that it completely removed this improvement on the prediction of non-spatially tuned neuron responses. And so the next question is then, why those nonlinear recurrence would matter for non-spatially tuned neurons? What is the link between those two? Well, previously it was hypothesized that the numerous recurrent connections in hippocampus could be critical for storing and recalling associative memory. And so next we look at the relationship between spatial tuning and memory processing. And what we saw is that neurons were less and less likely to exhibit spatial tuning as they encode information from further in the past. So you can see here that perceptual neurons that encode currently perceived information are very likely to have some spatial tunings, but the distant memory neurons that encode information from the past are very unlikely to have those spatial tuning. So together it would suggest that this, as information is a process over longer and longer time steps, those spatial tunings are more and more likely to be obscured and create those two different neuron profiles that are not really about spatial and non-spatial processing, but about the linearity of these processes. And so it creates these spatial tuned neurons that linearly encode the perceived information and the non-spatial tuned neurons that non-linearly encode past information. And the existence of those two types of neurons also suggests that the neural mechanisms that enable the hippocampus to learn the association between those stimulus that are separated by various spatial and temporal scales could be those two types of neurons, where there's the neuron that encode the features that need to be integrated, and the other neurons that encode the previously integrated information, so that the both information is available at the same time in the hippocampus, and that the new experience can be integrated with the previous experience. So that's it for me. Thank you.
Thank you, Maxime. Our next speaker is Dane Malenfant. Round of applause.
Hello everyone, my name is Dane Malenfant and I'm a computer science at McGill University with Blake Richards. And I'm also a citizen of Métis Nation, Saskatchewan, one of the Indigenous nations in Canada. And this nation, we have this cultural system called reciprocity, and I define here as traditional reciprocity, as like the expectation of leaving unneeded items for others who will do the same for you. And this kind of reciprocity used to exist in these shrines called Manitokan that existed on large ranges in the prairies of Canada. And they would have food, medicine, tools for people on long distance and traveling if they need something, but there's this expectation that you give away things you don't need. And what we wanted to do was kind of consider this behavior that exists in humans and animals and try to model this with reinforcement learning. And so I decided to try to treat this as a credit assignment problem. And credit assignment is the decisions that contribute to the maximization of reward or success. And so what I did is create this novel environment called Mashtan. And Mashtan is a mini-grid environment, and essentially the goal is that every agent has to learn to find a key and open a door, and it receives an individual immediate reward. And this is a very classic test in credit assignment. And what agents have to do is learn when to find a key and when to open the door when they have it. And this is a form of temporal credit assignment because they have to associate this in a limited time frame for certain time steps. But unique to this environment is that if all agents are able to open their doors, there is a large collective reward that is propagated to every agent that's larger than the immediate reward. And this is an example in multi-agent as structural credit assignment. And essentially, the key action that agents have to learn is to drop the key. But I ran through many different architectures, modern ones as you see in this graph, and the maximum success rate is about 28%. A lot of the algorithms that were invented in the last 10 years that try to do this form of structural credit assignment, like value decomposition and the various proximal policy optimization algorithms, they don't go better than about 3% or they completely collapse. The only one that showed any form of performance at around 28% is independent policy gradients. And this policy gradient algorithm was originally invented in 1992, and it was very surprising to see that it had such high performance in this sparse reward environment and was able to sometimes associate that this large collective reward is able to be received if the agents drop the key. And so I would like to leave you with this kind of idea of what I'm trying to tackle, and it's as agents are increasingly deployed in our societies, why can't they drop keys? And these graphs on the side I've shown as a little bit of an inference on the underlying learning dynamics that agents are incapable of achieving this dropping the key. If you look at the top graph, it's a histogram of the time step that agents in the QMix algorithm are able to receive their reward by opening the door. And what you see is that agent 2, it's very left skewed and it's able to minimize the time steps it's able to get to that reward. But as we know from the previous graph, QMix collapses. It's in the green one at the bottom. When you look at independent policy gradients on the bottom graph, you see it's more uniform and you see that these agents are more coordinated. Yep. Thank you.
Thank you, Dane. Up next is Serena Wang.
Hello everyone, my name is Serena, and today I'll talk about our work on real-time detection and in-phase stimulation of slow oscillation and sleep window. So perhaps you have thought of improving your sleep improving your memory during your sleep, and this is exactly what we want to work on. So we are particularly interested in two types of wave. One is sleep spindle, which happens in non-REM stage 2, a relatively fast frequency. Another one is slow oscillation, which hallmarks slow wave sleep. We also call it deep sleep. And as the name implies, it is slower with a larger amplitude. So these two waves are implicated in memory consolidation. We know that deep sleep helps with growth and help with recovery processes, and information processing happens exactly in the up phase of the slow oscillation. And it's also in relative to spindle activity also during that window. So it will be very interesting if we can interact with this wave, and then if we can increase the frequency, this could bring potential benefit to memory or even our immune system. And it can also help us to study the underlying causal relationship between some brainwave and our cognitive function. So closed-loop auditory stimulation is the exact technique that allows us to do this interaction. So by delivering auditory stimuli, which is like a sound, at the zero phase, at the peak of the slow oscillation, we are able to modulate brain activity, increase the frequency or amplitude, thus enhancing memory consolidation. So here it's important to note that we want a tool that can target any phase of any oscillation. This is on one hand for maximum experimental flexibility. On the other hand, research have shown that you need to target at a specific point for the maximum effect. So let's say if you do it at the down phase of the slow oscillation, there will actually be no positive benefit. So we work on Portaloop. We designed this small deep learning-based open science tool. It's specifically for closed-loop brain stimulation. So there you'll see the general workflow for Portaloop. So we are now using EEG, but you can use other data, and we're doing auditory stimulation, but you can do other like magnetic or other sensory stimulation. So this is portable, it is quite small, it's low power, and we have previously validated our work of spindle detection with it. So we have our own RNN-based detector. We also employ some continual learning technique in which we adaptively choose parameter for test subject during the night. And we are able to do both detection and stimulation in 200 milliseconds. So this proves that Portaloop can achieve this kind of low latency simulation, which is pretty good. So now we would like to add the ability to also detect slow oscillation and to do in-phase stimulation for the two waves. So let's split it up. Firstly, we would like to do a real-time detection for slow oscillation. So here the challenge mainly comes from a lot of delays. The delays that comes from online filtering, also from the software and the hardware delays. But our algorithm is able to achieve accuracy of around 0.86. So after the detection, we'll move on to in-phase stimulation. So stimulation, researchers often use some non-adaptive method, which means they would choose a fixed parameter for a test subject for the entire night. So it's fixed, it's hard-coded. As you can imagine, it's not as optimal because you have different brainwave amplitude. There will be a small variance there. So here we choose a more adaptive method, which is to use phase-lock loop. So phase-lock loop, in essence, is a feedback loop system which involves an artificial oscillator. So by comparing the input signal and the output of this oscillator, we continuously update the oscillator such that we can track the wave of whatever we are interested in. So here we're trying to track slow oscillation, and we're able to achieve a much lower phase error. So now what we are working on is actually to reuse this phase-lock loop and to use adaptive parameter for spindle. In the end, we would like to use the detectors and stimulators with Portaloop on different test subjects and to understand the simulation effect of conjunctive and disjunctive period of the two waves. Thank you.
Thank you. So up next, please welcome Darius Vilevicius.
Hi, I'm Darius, and I'm in the Eclipse Lab. I'm a PhD student at University of Montreal, and this is some work we've been doing on evolving stimuli for affective neuroscience. So if you've seen the Harry Potter films, you might be familiar with a a Bogart, which is a magical creature that transforms into your worst fear. And so what we've done is we've basically created this with a combination of brain decoding and generative AI. So why would we do this? Well, the general idea is that a big limitation in our neuroimaging studies is that we're limited to correlational analyses. So for something like fear or other conscious emotions that we like to study, we can only see what brain signatures are correlated with our response variable. But it would be interesting to be able to target or derive some pattern of neural activity to see if the person responds the way we think they're going to respond, or in this case, to see if they produce the type of image that we think they should be targeting. So this is inspired by some pioneering work by Carlos Ponce, where they used generative AI and intracranial recordings in macaques to drive the activity of certain neurons in the upper visual stream. And we've basically ported this approach to whole brain human fMRI. So we've run a pilot study to start with to test out our image evolution paradigm outside the scanner. So we used a couple of different measures to try to drive evolution of images in this animal subdomain of CLIP. So we used self-reported measures of fear and skin conductance responses. And we found that for both of these metrics, within 8 to 10 generations, the images converged on similar areas of this latent space, on average across people. And it converged where you would expect on things that are generally feared by most people, spidery things, lots of legs, different kinds of buggy-looking creatures. And in our control condition, where we used instead random fitness values to make sure it wasn't some kind of random drift, we found no such convergence. So now we're bringing this to the MRI. So we're piloting the fMRI version of this experiment. I've been playing around with within-subject brain decoders, and those seem to work pretty well so far. But one use case that we kind of stumbled on recently was the possibility of downloading some brain signature or some beta map from the internet, for example, for fear or for something else, or pulling a neurosynth map and using that as a target and seeing, well, what does it produce when we try to select for activity in this network or this region. So as a quick example, again, we're just piloting, so this is one run, not necessarily conclusive. But if you pull the neurosynth map for fear, it's basically bilateral amygdala. It's quite constrained, which Luis Pessoa might have a problem with. And we found some kind of unexpected result where when we try to maximize for activity in this region, we got not things that are scary, but instead these cute little fluff balls. So it's kind of an interesting potential method to probe neural signatures and different kinds of brain metrics. And if you want to hear more, you can come see me at my poster 51. Thank you.
Thank you, Darius. So up next, we have Jan Lezang.
Oh, sorry. I don't think this is my presentation. Right. Sorry.
Apologies, Yanle. If you're down for a switcheroo, we'll welcome Miriam Sarawi.
Okay, so good afternoon, everyone. I hope you've had like a great conference so far. So I'm a PhD student at University of Montreal, and I'm working under the co-supervision of Karim Gerbi and Bruno Gauthier. And today I'll be talking about a study we've just conducted, which is the data-driven exploration of neurotypical children using the 5-point test. Cognitive profiles, sorry. Okay, So we all know that executive functions play a major part in children's development. They've been associated regularly with future academic achievement and the development of other cognitive abilities. Inter-individual differences are also well documented in children, specifically young children. Unfortunately, only a few studies have explored different executive profiles in neurotypical children. And with the new opportunities that are offered to us with the development of machine learning and new executive functioning tests, the goal of this study, the goal of this study was to get a data-driven exploration of our executive profiles, so using the 5-point test, which I will tell a little bit more about. So in this study, 113 participants were included, 7 to 13 years old. And they've all participated in the 5-point test, so, and specifically a numerical version of the 5-point test. So the 5-point test is a design fluency task, and it consists in asking the participants of drawing the most different designs possible in 5 minutes. So this is an example of design that could be drawn by participants, and then they would press the arrow and go to the next to draw another design. And so we used the number of designs produced, the number of repetitions, and the number of strategies across 5 minutes. And so for the results, sorry, there's a problem here. Okay, so I'll just do that. Okay, and so for pipeline, I'm sorry, I have a little mistake, but we did 2 steps. So our first steps was to use unsupervised machine learning to get the clustering of the profiles. And the second step was to then use a supervised machine learning algorithm to try to understand a little bit more the structural aspect of those clusters and what differentiated them. And so 2 clusters were separated with a silhouette score of 0.33. So that's tend to be overlapping clusters, but still different ones. And so both algorithms gave the same results. They were pretty similar, so we decided to continue with only the k-means. And so we applied a logistic regression on the k-means to explore the future importance. And so, as you can see with the red rectangle, the most important features were the design at minutes 1 and 2, and the number of strategies at minute 1. And so both algorithms identified 2 distinct profiles, and those profiles were different under protection and use of strategies, which we think are associated with cognitive flexibility and working memory. I know it's a little out of nowhere, but I'll explain why. So basically, the 5-point test has been mostly associated with cognitive flexibility and working memory. And so we believe that the use of strategy in the beginning of the task would be a witness of the participants' ability to think outside the box and to try different strategies. And for the production, since we've had, since in the task, you cannot see the previous design. The working memory is used there. So the participants were able, in the beginning of the task, the first group, to produce way more designs. And so, yeah. For future steps, more studies are needed for better understanding the source of these differences. And we're going to start a project on exploring the neural basis of those profiles using mobile EEG, specifically the DSI-24. And so we're really excited. So it's going to be the same pipeline, essentially, but with the synchronization of the DSI-24 during the 5-point test. And so if you want to know more, well, thank you very much, first of all. And if you want to know more, you can come see me at my poster, poster number 43, Tuesday and the next day. And I would like to thank, of course, the conference, everyone, the organizers, and also both labs, my labs, so the COCO lab and the Linea. Thank you.
All right, so next up, please welcome Alejandro Salinas.
Hi everyone, I'm Alejandro Salinas Medina, and I will be talking about the neuron level analysis of the big brain. Neuroscientists have studied the fine distribution of the neurons across the cortex with what we know as cytoarchitecture or cytoarchitectonics. Here, precise quantification has been long appreciated, especially now that we are able to explore the brain in ways that are yet unseen details. Classical analysis rely on pixel level analysis and textural features, but these offer low interpretable data. In this research, we aim to move from pixel-level analysis to cell-level analysis. This is enhancement because cell-level analysis offers a more biological, meaningful interpretation dataset. So pixel-based analysis work with raw image data that are, some examples are intensities and spatial patterns. And this does not offer any kind of biological interpretation. On the other hand, cell-based tissue characterization focuses on segmenting and identifying, segmenting, and analyzing individual level neurons and its properties. So this allows us to move from a pixel level analysis abstract data, image data, to a more concrete biological interpretable data. So for this, we present this novel pipeline that has been totally powered by GPU, where as an input it receives big brains section with 1 micrometer resolution. Then it runs a partial differential equation based cell detection in order to mark the centroids of each of the cells on the big brain slice. Then we perform a cell segmentation that currently we are working on benchmarking different kinds of cell segmentation algorithms. And then we apply a morphological feature extraction from the segmented cells. And we run this for different number of big brain slices, and then we stack them together in order to have a 3D volume segmented cell data. So our pipeline has been able to create cellular level representation by automating a large-scale generation of tissue features, and it's scalable and GPU-friendly. So the segmentation we're seeing there, it took 4 seconds to run and to segment all those cells. And this will produce a suitable data set for machine learning tasks, and that has a more biological, meaningful interpretation. For the next steps, we have automatic cortical layer segmentation using this data set that has been created from cellular level segmentation. And this comes from highly interpretable data. And well, we are aiming to create a data-driven, observation-free, and large-scale analysis. And we are also aiming for more high-quality and high-resolution images. Thanks.
And finally, please welcome Yanlei Zhang.
For some reason, my PowerPoint is not in there, so I'm just going to plug into my computer. Sorry about that. Sorry about that. All right. My name is Yanlei Zhang, and I'm a joint postdoc, joint affiliated at Yale and Mila. So today I'll introduce a framework for learning time-evolving neural signals using the geometric scattering and homology framework. So this is joint collaboration from Yale, Mila, and Duke and Columbia. So just for the method, I'm not a neuroscientist, but just for the method, so we're interested in how connected the brains are in specific tasks. So we want to understand if there's a way to characterize time-evolving signals in the brain in loaded dimensional space in such a way that it is interpretable and is useful for downstream tasks. So given the whole brain, the time-evolving signal can be extracted using passivation. So in return, it gives spatial coordinates of the parcels and also the time-evolving signals at each vertex. So here we can construct graphs out of these parcels. So the graph can be constructed by a graph kernel using different bandwidth, and subsequently we can have a set of graphs and each corresponding to a construction, a connectivity of the brain. So in this case, the vertex represents a parcel, and then the edges represents how strongly connected each parcels are in specific tasks. So we all know that in spatial domain or vertex domain, the signals has certain frequencies. So in order to extract these frequency information, we're going to use analog similar to a Fourier transform, which is scattering. So in scattering, you have these wavelets where the wavelets will collect the frequency information, and then subsequently gives you the existence of frequency in the vertex domain. So in that case, we use the learnable scattering. So basically, your wavelet can be learned. So it won't be restricted to a snapshot of a certain wavelet. So then wavelet coefficients can be collected here, noted as Sx. And then, so this is still in the high dimensional, so we want to understand, neural signals in lower dimensional space. Therefore, we're trying to learn a lower dimensional representation. So in this case, we use the autoencoder. So here we have two parts. The first is the encoding part, represented by E, and then decoding part represented by D. So this latent representation can be regularized incorporating different geometric informations. So in this case, we're incorporating fate informations. So fate captures both local and global geometry simultaneously. So different than its counterparts with UMAP and t-SNE and other methods. So here, T-fate is incorporating the time lapse into play of the game. So this representation then can be separately used for analyzing the brain activity by path signature, persistent homology, or just simply an MLP classifier. So, so just three examples to demonstrate the validation of this method. So the first one is neural model coupled oscillator. So this mathematical model is usually describe the collective behavior of coupled oscillators. So in this case, we simulate 64 coupled oscillators. So there are 3 rows of these grids, and each one is corresponding to the different frequencies. So here we have each node or each vertex have a natural frequency denoted by omega, and then the coupling strength gives you the synchronization of the system. So the first row is the weakest coupling. In return, that this system won't be synchronized at the end. Therefore, the neural GSTH trajectory, as you can see, probably too small. So it distributes an equal distribution with time. Therefore, it doesn't converge at the end, whereas the intermediate coupling strength actually at the end converges quickly, whereas the last one, which is full synchrony, and converges very fast at the end. So similarly, this can be seen from the zeroth homology of the Betty curve. So the Betty curve actually characterizes how fast it converges at the end in terms of the time lapse. So the second experiment is done on the mice. So basically, in the front, you have the biphoton projection of lights, and then the mouse is reflecting to these sinusoidal signals, and then these signals can be collected using calcium image signaling. And so neural GSTH correctly recover the patterns of sinusoidal patterns that's recorded in the mice of the brain, in the brain of the mice. Sorry. And the last example is for schizophrenia patients, that it is well known that schizophrenia patients are not usually very sensitive to emotions and people's facial expressions. So in the setup of this experiment is the subject is asked to observing an actor, which is looking at different kinds of movies, for example, horror movies or movies with cats, and then the subject is going to observe the facial expression of the actor, and then the Fnir's signals are collected from this observation. So this observation has 3 periods of time. The first one is movie watching, second one is directly in eye contact with the actor, and third period is resting state. So neural GSTH can correctly identify the switching of the task, which in particular the first trajectory, you can see that when the task switches, and then the trajectory also switches direction. So our model also correctly or in high accuracy recovers the classification error of these tasks. So my poster is at 57, so if you want to know more details, come to talk to me. Thank you.
Thank you to Yanlei and everybody who presented during the lightning talks. If I can ask you for your patience for one final moment, Karim Gerbi shall address us all before we move to the Hall d'Honneur for the reception.
Thank you so much, Sean and Charlotte, and I would like to thank the SAC for organizing these lightning talks. There's another one tomorrow, as you know, and it's always so exciting to see all these projects that are being led and all the teams and all the students that are working on these projects. So it's been a nice way to end it's not over yet, but it's a nice way to end the presentations of the day. So please join me one last time in thanking all the students that presented this evening.